is Northwestern Agricultural and Forestry University in Yangling. And he, right away, we could tell this was an unusual person. Worked very, very hard, so hard in fact that one of the postdocs pulled me aside and said, "You have to tell him to stop." And so I tried, but he has really been a tireless, creative researcher. He's worked on a huge diversity of projects, some of them very challenging, most of them very challenging. And it's really exciting that now he has too many projects to present, so we've been working on simplifying it. So he also is quite an adventurer. Probably he's visited more places in California than any graduate student I've ever had. And he's been just a joy to have in the lab, and we're very proud of your achievements and excited to watch your career. So thank you.
Thank you for the introduction, Pam. And good morning, everyone. Today, my title for the topic is molecular characterization of rice pattern recognition receptors interacting with the Pseudomonas aureti passiva aureti infection machinery. And during my talk, I will use XO, stand for this bacterial pathogen. So, I came from China in 2012, and I was a master student in China. I used to work on the wheat strep rust, which is a severe disease in wheat causing yield loss. It's a fungal pathogen. And then I joined the Robert lab team in 2012, worked on the bacterial blight, which is the bacterial pathogen. It had one Flagella, one bacterial pathogen, and it also caused severe yield loss in Asian countries. And African countries where rice is the main crops there. And in 1997, former lab members in the Ronald lab identified the first plant pattern recognition receptors, the plant PRR21 in rice, which confers a robust resistance to a lot of XO strains, and it has LR domain outside of membrane, transmembrane domain, and the kinase domain. And we know, in the floor gene for gene hypothesis, if there are gene in the plant, there should be a aviar gene or some effector or PAMP, which can be recognized by the PRR, then trigger the immunity. And then my first project, working with the former postdocs in the lab, we try to identify this gene, which is from the pathogen XO. And then finally, the postdocs in the lab find a small peptide we call pep protein called RAXX was secreted from the type 1 secretion system in the XO. And when it was sulfated by another gene called RAXFT, the functional version of RAXX can be recognized by 21 and trigger the immunity. And the people find in the lab find the loss first. And the PR proteins are very good marker for the immunity response. And this is the four parts of my talk today. So the first part is the variation of the Pseudomonas genes that are required for the activation of actin-1-mediated immunity. So our collaborator in India found the field strains from India and also Africa. These are field strains, but they invaded the actin-1-mediated immunity. And I'm eager to find the mechanism. What's the reason these natural variations of strains can invade the actin-1-mediated immunity? So as I just presented RAXX, we confirm it's a very important gene secreted from XO. So the first hypothesis is we can compare the sequence of the two strains, one from India, one from Africa, with the PS9 RAXX. So you can see there are 10 of the SNPs change the amino acid. And the former paper we published in 2015 already proved that the proline 44 and proline 44A, the two sites are very important for the functional RAXX. And you can see the two strains have the amino acid change in the proline 44 and proline 48. To support a prove this hypothesis, right? So I'm trying to clone the RAXX functional one from PS99 and use a plasmid to transform it into the African strain and the Indian strain to see if I can restore the phenotype. So these are typical lesions and assays we use for to determine if the strain is kind of virulence or virulence on the plants. So we use like a bacterial inoculum, dip it in the scissors, and then inoculate the top of the plants. And 14 days later, we will measure lesions on the leaves. You can see the resistant one, there's like no significant phenotype. But for susceptible leaves, you can see the very long water-soluble lesions. And this is the assay I'm doing, so I did the inoculation. So in I-21 plants, you can see the wild type PS99 is resistance. And if we knock out Rex X, it becomes susceptible, and we can complement it by introduce the Rex X again. So I try to complement the African Indian strain. So I put the PS99 Rex X into the Indian strain. You can see I can restore the phenotype. So in this strain, the data suggests that the Rex X dysfunctional Rex X in Indian strain is the reason why this strain invaded the H1-mediated immunity. So I did the same thing in the African strain to say it's the same case, but interestingly, I cannot complement it with the PS99 Rex X in African strain. And if I knock out the PS99 Rex X, I'm trying to use the African strain Rex X, it also cannot complement the phenotype, which suggests that the Rex X from this African strain is dysfunctional, but it's not the only reason why this strain invaded the H1-mediated immunity. Then in the exogenome, Rex X is here, and Rex T is close to it, which are the tyrosine transferase, which are sulfated Rex X to be functional Rex X. And Rex AB is close to it in operon, which is the type 1 secretion system secreted Rex X. So, and previously, we already published paper in 2004 in MPNMI confirmed if we knock out Rex X, the strain will become virulent. So I'm thinking maybe Rex T is also not functional in African strain. Just part of my hypothesis. So I just tested if I use the African strain Rex XT to complement the PS99 Rex T mutant, but I cannot complement it. But if I use the wild type Rex XT from PS99, it can complement the phenotype. So which suggests the Rex XT from African strain is also functional. So as I mentioned, this two genes is very close in the genome, and they might be share like a potential promoter. So I clone the whole genomic DNA piece, include the two genes, and use the two gene whole genomic DNA piece to put it into the Rex X mutant, and also in African strain mutant to suggest my hypothesis. These two genes are the reasons why the African strain invaded the immunity. So finally, I can see in the African strain, if I put the Rex X and Rex T, the whole genomic DNA piece from the wild type, PS99, I can complement the African strain. So which suggests that my hypothesis is correct. So both the Rex X and Rex T is functional in triggering H1 immediately. Then I compared the genome sequence of the gene sequence from the PS99 Rex T and African strain XT, and I also found several SNPs, and seven of them changed the amino acid. So I tested them one by one, mutated them to test which one of them is the reason makes the Rex T dysfunctional in activating H1-mediated immunity. And based on my result, the H50 and R109 is the two important sites that determine the function of the Rex T. And this is a figure from one of when we submit the paper. They give us this model, a homogeneous model to suggest why these two sites are so important for the Rex T functional. So Rex T is a very high homology to human TPS-T2 gene protein, which also is a sulfotransferase. So they built a model based on the TPS-T2 model. So you can see the Rex T H50 is in the interface between the two monomers of the enzyme, and it could be affecting the dimerization because in the TPS-T2, if they mutate the similar place here, it makes the protein loss the function. And for the Rex T R109, so the Rex it's the phenotype is likely due to a disruption in the binding of the Rex T peptide. So this is the peptide substrate for the TPS-T2, and the Rex T is close to like the peptide binding site. So maybe it's the reason why these two sites is so important. So conclusion for this part is the first Rex X in the Afro-Indian string is dysfunctional for why this string invaded the actin-1-mediated immunity. And both Rex X and Rex T are the genes of why the Indian African string is dysfunctional for activation of actin-1-mediated immunity. And the two sides I just showed is the reason why the African string Rex T is dysfunctional in triggering actin-1-mediated immunity. So we'll move to the part 2. So from part 2 is the more projects I working on the rice, the host side. So I mentioned about actin-1 is the first PRR we identified in the plants. And in 2004, a Chinese group found another plant PRR. It's called XE3, which have like a very high homology to the actin-1. It also have like a LR domain, the memory domain, and a kinase domain. And we have to find many regulators of actin-1-mediated immunity, but the last was studied in XE3. So I'm thinking maybe we can cross several of identifying the components in actin-1 to test if any of them also shared by XE3 signaling pathway. So first, because all the actin-1 study was carried out in the Kitaki variety in rice. So first, with the former postdoc Wei Guozhang, we transformed the XE3 in the Kitaki background, and finally we selected two of the homologous homozygous lines which have contain XE3. You can see I can detect the protein by the UBI XE3 flag tag line, and all of them is resistant to the exo string TSO79. So as I mentioned, like if we have identified several of the regulators of actin-1-mediated immunity. So one of them is called OSF2, which is a co-regulator of actin-1-mediated immunity. It has the LR domain and transmembrane domain. It's a positive regulator of actin-1-mediated immunity. So I want to know if this protein is also a positive regulator of XE3-mediated immunity. So then I cross the XE3 line with the OSF2 RNA line in the target background. And in the F2 population, I select the lines both contain the XE3 and also contain the OSF2 RNA. So and inoculate the plant. So you can see the plants in F2 contain both transgene is susceptible, which suggests the hypothesis that OSF2 is also required for XE3-mediated immunity. And XB15 is identified as 3:2 hybrid in the lab. Like several years ago, it's a negative regulator of the actin-1-mediated immunity. So through the same assay, I did a cross. And in the F2 population, the plants contain both XE3 and XB15 overexpressed. So all the plants are resistant, which suggests that XB15 is only required for XE21 signaling pathway, but not for the XE3 signaling pathway. And the same thing with XB24, which is ATPase, which is also a negative regulator of the XE21 signaling pathway, and I did the cross. And in the F2 population generation, all the plants contain XE3, and XB24 was expressed, they all resistant, which suggests this negative regulator is not required for the XE3-mediated immunity in the Kitaki background. So until now, the data from this part, the first conclusion is the rice gene XE3 confirm robust resistance in XO, also in the model rice Kitaki. And genetic analysis results show that OSF2 is also a positive regulator in the XE3-mediated immunity, but these two are not. So now we start, we already, and as I mentioned, we already identified OSF2, XB24, XB15 as a regulator of X21-mediated immunity. To identify more of regulators in the XE21 signaling pathway, several years ago, Dr. Moshen Chen in our lab generated a fast nutrient population in the UBI X21 Kitaki background. And we grow the mutant line in the growth chamber, and we inoculate the plants. Because as I said, the background of the plants contain X21. Supposedly it should be resistant. But if we mess up or disrupt several important components of the signaling pathway, the plant will become susceptible. So we identified about 10 of the lines in which actin-1 mediated immunity, and we send the lines with the collaborator in GFA and do the Illumina sequencing to identify what kind of mutations caused by the fast mutation in each line. And during the same time, we back-cross the susceptible lines with the parent line, you get one Pitaki. And in F2, all the mutation is segregating, and the phenotype is segregating. And combined with the sequencing data, we will try to identify the genes which is candidates affect the immunity. So one of the line I'm working with is called SEXY4, which stands for the suppressed or actin-1 mediated immunity 4. So you can see the same inoculation assay data. So Kitake wild type, there's no actin-1, so it's fully susceptible. And the UV actin-1 Kitake is resistant. And the SEXY4, you can see it's also fully susceptible, similar to Kitake, which stands some gene is messed up in this mutant line. So this is the F2 segregation data I just mentioned. So we are trying to use the PCR to detect all the mutations based on the sequencing to see which one of them is segregating with the phenotype. So you can see this is detecting one of the mutation. I will tell you later. So all the plants contain this transgene, they are susceptible. And the ones do not contain the gene, they are resistant. And this is 3-to-1 ratio, it's just that it's a dominant mutation. And further analysis with the help with Rashmi Jain in our lab and Botian, and we identified that there are 32 kb translocation from chromosome 3 inserted into chromosome 7. And there's a 4 gene is contained in this DNA part, and this gene on the right border is rice dicer-like protein 2A. And first we are trying to test if any of the gene, the expression level was affected. So based on the qPCR data, you can see the gene on the right border, the dicer2, is kind of 2 to 3-fold high expressed in the mutant compared with the wild type, the parent line. And there's one gene is a pseudo gene, not expressed at all. And then we are trying to find why this gene, only this gene was affected. So a close analyze find out like the gene, like the prime UTR and the whole CDF, ORF was still there, but a part of the 3' UTR was truncated during the translocation. So I'm thinking maybe the part of the UTR is important to regulate the gene. And we also did the silencing this because in the SEXY4, I told you like the dicer2 is a 2-fold, 2 to 3-fold high expressed. I'm trying to confirm this gene is the candidate for the mutated phenotype. So I try to silence the gene with RNAi in the mutant background. So this qPCR data confirming the T1 generation, the gene is silenced even lower, a little bit lower compared to wild type. And after I inoculate the plant, you can see the silence line, the resistant phenotype was restored, which suggests that a true gene overexpressed is the reason why this plant becomes susceptible. So as I mentioned in the SEXY4, the UTR region was truncated, only remaining 120 base pair, but the full length of the 3' UTR of the gene in the genome was 477 base pair. So I try to test my hypothesis. So first one is I fused the different part of the 3' UTR, like 477 is the full length of 3' UTR, and the 120 was like what's left in the SEXY4 background. I infused with the EGFP and to test in the rice plot to test the signal of the EGFP. So you can see compared with the full length, the truncated one, the EGFP signal is way much higher compared with the full length. So we just suggested my hypothesis. It is because of the truncated UTR. There are some sequences repressing the EGFP expression in this region. So then, my further question was, what's the mechanism? Why the susceptible? Why the phenotype is susceptible in SEXY4? So is it actin-1 directly was affected or something else? So first thing is we detected the H1 protein and the RNA level in the mutant. So you can see the RNA level is significantly lower in the mutant and was restored in the silencing line. And the protein is also like it's H1 protein with some Western blot. So you can see Kitaki there's no H1, and compare with the parent you get H1, 64 there's less protein, and protein level can be restored. So H1 was restored, but we don't know if this much level of H1 is still enough to trigger the immunity. So as I mentioned in the beginning, we have like ROS1 PR gene induction, which is like a commonly used like a immunity marker to confirm the signaling of the immunity is still triggered or not. So this is like a ROS1 after treatment the leaves of the like a wild type and UDF1 and 64 mutant treated with the REX peptide. So you see in the Kentucky, like either treated with MOG or other sulfated one or non-sulfated version of REX, there's no ROS1. But in the parent line, UDF1 in Kentucky, you can see with the functional REX sulfated peptide, there's strong ROS1 trigger. And then in 64, interestingly, the immunity was gone, but somehow the ROS1 is still there. And the PR10B is like a food marker gene in rice, also in Arabidopsis, it's like an indication of the immunity response. So I also the same assay treated the leaves of the UBM1 Kentucky and 64 with functional REX. So you can see in different 3, 6, 9, 12, 18, 24 hours of treatment, the PR10B is still existing in 64. Might be a little bit lower, but there's no significant difference. And I joined the summer school in Simsbury last year. I talked to people there, like it was said, oh, maybe the mechanics is a very good important pathway in the Arabidopsis signaling signaling. So I spent like 3 months set up the assay in rice, but finally there's no difference. So you can see in the mock treatment plants, there's no like no mechanism and phosphorylation can be detected. So this is an antibody from the human and it's widely used in like plants, animals. You can detect like a phosphorylated mechanism 4 and 6. So in the mock treated like all backgrounds there's no mechanism. And Kentucky is no with the treated XX, but you get one that is strong signaling. And in 64 mutant, still same. So not mechanism either. Then at the same time, with the help with the former postdoc Tony, and we did the RNA-seq in the line to see if we can find more candidates which are important why the plants is susceptible. So you see this is the top 10 like a different expressed gene. The first one is the ritual transposon, and the second one is dicer, which also suggests our data is correct because dicer is overexpressed in 64 mutant. And the third one is called a Burke domain contingent protein. And there's no so much study about this gene, but none of the rest either. But I look for papers published in rice, and they found one of the Burke gene actually was one they used it as a marker gene once the plants, rice plants, was treated with the rice blast fungus. This gene is highly expressed, used as a marker gene. So I'm thinking, oh, maybe the gene have something wrong in the signaling. So I use the CRISPR-Cas9 to mutate this gene in the UBM1 Kentucky review resistance into the transgene. So these are two independent T1 populations, and they have different mutations of the Burke gene in the genome. So this one is a biallelic, has either 253 base pair deletion or 6 pair deletion. And all the plants are susceptible, and this is another independent population. They all have like a 1 base pair insertion in the targeted site to treat the frame shift. So both populations are susceptible. So we just suggest Burke gene is kind of important in the H1 immune signaling pathway. So conclusion for part 3 is first one, the 3' UTR of the dicer2 contains negative regulatory elements that controls the dicer2 expression. Second is the roster, mechanism activation, and the PRG induction. Another compromised in SEXY4 mutant. We suggest in SEXY4 there should be some other signaling pathway was affected. So it might be the BERT. If the BERT is down-regulated in SEXY4, and then if we muted the BERT in the wild type, the UBI21 plants, the plant becomes susceptible. So the last part of my talk is a study of another sexy mutant in SEXY5, collaborator with the former postdoc Wei Wozhang. We found another gene called OSSKI2. It regulates the SU21 mediated immunity to XO. So we did the same thing, similar as SEXY4. So you can see the inoculation. SEXY5 is fully susceptible, and this is the co-segregation assay as I showed in SEXY4. And in this time, it looks like it's a recessive mutant. Like the resistant and susceptible is 3 to 1. Three ratio plants are still resistant. So with the co-segregation assay help with the rationing, we find like at this time there's a 843 base pair insertion was inserted in just after the start codon of this gene, and I used the qPCR again to test if any gene 20 kb upstream and 20 kb downstream was affected. So this time you can see the gene, the inserted gene, this one was totally knocked out in the mutant, and all other genes downstream, upstream, there's no affection. So kind of suggests that, oh, this gene might be a candidate. So then we did the complementation. So I amplified the 14 kb gene of the genomic gene sequence that contains the promoter of the gene and transformed into the SEXY5 background. So we can see the teaser plants, like 10 of them, or 8 of them, 6 out of 8 were restored, and only line 5 and line 7 not restored. And I tested them with qPCR detector. Like this lines, there's no expression, which makes sense. Like this gene cannot express and not restore the phenotype. So this complementation suggests that the OS2 gene is the candidate gene. Why this SEXY5 mutant is susceptible. So then we detect the actual protein to see if it's affected or not. So this is like T1 population, like all 12 plants, the 3 of them do not contain the transgene are susceptible, all others contain the transgene, they are resistant. So and in SEXY5, you can see the XEPI was put in a knockout, and also H1N1 was like significantly lower compared with UBF1 Kitaki. And both genes was restored in the complementation model. And we detected protein level, so with the antibody detect H1N1, the detected H1N1. So Kitaki is no H1N1, and in the UBF1 there's like strong signaling of H1N1, and in SEXY5 there's no H1N1 at all. And it can be complemented in complement line, which suggests that SU21 protein might be totally gone, and this is the reason why SEXY5 is susceptible. And I will detect the two good markers for the immunity response, the ROS1 and PR10B. So you see the mock treated plants, the mock treated plants, Kitaki, H1N1, SEXY5 mutant, and the complement line is no ROS1, and the non-sulfated Rex X has no ROS1, and only the sulfated Rex X in the wild type, the UBF1 Kitaki, there's strong ROS1, and in the SEXY5 mutant, this was gone, and ROS1 was ROS1 shows again in the complement line, and also for the PR10B induction. It was gone in SEXY5 and it was restored. So in SEXY5, like H1N1 protein was gone and the ROS PR10B are gone. So this is a good model published in Science in 2015. So they studied the SPI2, which are RNA helices in the Arabidopsis. So their model is if there's an aberrant coding transcript, so SPI will degrade the RNA from 3' to 5'. But if this RNA is dysfunctionally, the aberrant RNA will go to the RNAi or microRNA pathway will be degraded. All the aberrant coding transfer will be degraded by either dicer2 or dicer4 in the Arabidopsis. And you might remember in 64, the gene I'm working on is dicer2 in rice. And so 65 mutant, my kind of gene is 65. So I'm thinking, how about if I knock out dicer2 in 65 genetic background to see if I can restore the phenotype, to see if rice and Arabidopsis have similar model in RNA decay. So I use the CRISPR-Cas9 to knock out the dicer2 in 65 background. So this is a two independent population. So in this population, there's like either 24 or 26 base pair deletion in the one plant. And this is another population, they have like a 30 base pair deletion of dicer2. And you see if I knock out the gene in 75, it will restore the phenotype. And also I can restore the RNA level of 21 or the protein level if I knock out dicer2 in 65, which is since dicer2 is kind of downstream of SPI2 signal capacity. So conclusion for the last part is the RNA helices OSNPI2 knocked out by a translocation was found associated with the recessive mutant phenotype in 65. And if I mutate dicer2, it will restore the resistant phenotype in 65. And in 65, not only the plant becomes susceptible, the plant also shows many like a development phenotype, like they are dwarf, they have less telomeres, they have like a lesion mimic in the leaves, and all the phenotype is co-segregating with the OS SPI2, and all the phenotype can be restored by knocking out dicer2. So this is the kind of hypothesis model based on my data. So as I mentioned at the beginning, XO secreted RexX, once it's already and secreted out from the PEP1 secreting system, it can be recognized by turning one into the right cell memory and treated immunity. And we always think that rosters, metakinase activation, or PR proteins are so important, but this is not the case in 64, so we don't know how important of these three markers in the actual mediated immunity. And whereas the SPI2 is RNA helices, and dicer2 is a kind of maybe downstream of SPI2, and dicer is known to germinate like a 22 nucleotide siRNA, so maybe some microRNA is directly targeted at work and affect immunity, and we are trying to confirm its work is so important. And based on the study, we find that work is kind of have polygalactoneurase is the kind of enzyme which can like digest the polysaccharide. So, and in the tomato, people find that during the fruit, like a ripen, to become like this kind of bird will like target the cell wall, so fruit will become more soft. But it's not the case in rice. So, and we know XO it has cell wall, and the XO will produce a lot of extracellular polysaccharide we call it EPS, and it's also reported as like a very important virulence factor that causes the phenotype in rice. So we are thinking maybe it's targeting either cell wall of this XO or the EPS secreted out from the XO. And finally, I want to thank many people, especially our PI, Cameron. Thank you for holding me in the lab for a long time, giving me so much advices and so much support. And I want to thank Dr. Moshen Chen, Benjamin, and Rory. This is like a doctor in our lab or former postdoc, giving me so much suggestions like how to analyze data, how to discuss with me with the project, and so many postdocs in the lab, and also Professor Valley. He helped me to write papers, give me so much suggestions, and offer me a lot of help. And I also want to thank people in the department like Han, Steven, Tyler, Amanda, and Isaac. They welcomed me very well in the first 2 years because there's not so much international students in the department. They invited me to the parties, soccer games, retreatments. I didn't feel like going to abroad. I feel like at home. Thank you very much, guys. And I also want to thank people in the department like Jessa, Maria. They're always very helpful when you involve them. And I also want to thank like meeting in my during my study, like Bryce, Ida, Dinesh, and Yuhong in my qualifying exam give me a lot of help. And these three professors, Bryce, Ida, and Dinesh, is also in my graduation committee. They give me a lot of edits and help during my writing the thesis. And Richard Bostock is my academic advisor. He always give me a lot of help and suggestions. Thank you very much.
Any questions? Student in the front row.
Yeah. Two, on the group.
Yeah.
But college electronics, does EPS have pectin in it, and do bacterial cell walls?
No.
Okay.
But actually, this one is not actually
It's not true.
It's just like a predicted.
Okay.
Yeah.
So,
And the R
cell
policy.
wall debris.
Yeah.
Okay. So, the other question, I forget, it is, essay 21 mediated removal is an essay, JA, both, or neither, depending?
I'm not sure, but I know like Dr. Moshin Chen is studying like assay pathway, the NPR1 homolog in the rice. I think he might be the answer, but I don't know.
Yeah, is there Naji rice? I think they were leading mimics.
Yeah, there is Naji, well, we can have Mashank answer that. There is Naji rice, right? But it's not so
Yes.
clear.
That's kind of an issue, but
But in rice, it's not so clear, it's not well separated, whether it's SA or JA pathway. It's not like in orthodoxis. And people use NAGI and find that it's not really involved in disease resistance, but rather cell death. So it's rice
it's
is not
rice.
like orthodoxis. Even though NPR1 has a conserved pathway, but the pathway itself is not like in orthodoxis. It's SA pathway, it's J pathway, well separated. In rice, it's not well separated.
So what does RAX-X do? Because when you're talking about these interactions and things, when you say non-functional mutant, that you're talking about non-functional in terms of interacting with XA21. What else? Excuse me, right? What else does it do?
So what it's doing in the axle itself, we don't know. Like Anna in our lab, the postdoc, he's working on the functional y-axis. What's the functional y-axis in the axle? But actually, he proved that it's kind of, if I remember correctly, she published a paper last year to confirm it's a virus factor in axle. Because if we knock out X and in other plants in the low density in the rice, it will like affect the virus. And also in the plants, we find out Anna find out like a Rex X, if we trigger it, if we treat this with the rice or adopted roots, it will promote the root growth, which is the mimic of plant hormone called PSY1. So yeah, also following up on the why it mimic like plant hormone and the promote growth.
This sequence is very similar to a conserved plant peptide hormone that's also sulfated. So that we think there's some kind of biomimicry going on, that it's producing and telling the plant to do something that promotes infection, but we don't know what that is. So we have a few people in the lab who's now working on that. It's a big question. Question in back? Did I see?
So actually, these two that university working as a actually to these kind of upstream regulators, not the whole
Yeah.
pathway. Obviously, we cannot link directly from the receptor to these two regulators. Kind of need to regulate the gene expression of this property.
Yeah, it's not like traditional regulator, just somehow if we overexpress or knock out the gene, it affects immunity. It's just regular, but not a traditional regulator. Yeah. Yes.
Did you check the protein level of XA21 in the birth mutants?
In which,
In the birth mutants?
I'm working on it right now. Okay, I just have the plans last week. I haven't do the rest yet.
So I have a detailed question about the experiment result. Actually, when you test the NMP activation in Rust system, actually, I only see one band. This is very typical in rice. Even in raw topic, usually we can detect three bands.
Yeah, in rice, in abduct it's like a traditional 3 or 2, I know. But in rice, actually, it's not well established in rice. And people can sometimes detect 1 or sometimes 2. So it may be based on like how it's experiment. Yeah.
So if you mix an Indian strain or Aspen strain with a regular strain, you're going to have different effects by themselves. But if you mix them together, do they have an effect on the plant that either benefits one or the other?
You mean like mix them and not the plants?
Correct, yeah, because I would imagine that that might be a natural situation that you have a mixed population perhaps of these
Yeah.
strains out there.
Yeah, that's a good question, but I haven't tested that yet. Yeah.
What would you expect? This is not a key.
You
But you
know,
may use the board.
actually, the kind of similar experiment, like because first when we identify RexX, it's created from the type 1 screen system. I mixed up like a RexX mutant, RexXT mutant, and wild type mixed them like a different ratio to confirm, if in the RexX mutant, in the RexXT mutant, it will secrete out the non-sulfate RexX. It might be going into the RexXT mutant, RexX mutant. The RexXT will sulfate this RexX and secrete out again, but the X doesn't work. So maybe like if I make up like a wild type and Indian string, then I'm like, yeah, I can test it. Yeah, if I can think about like a. Yes.
In the dicer 2 and birth mutants, did you check for like rice blast resistance, compromised rice blast resistance?
No, because I think like a XO like it's like an agent, and I think also rice blast you need like a permit to work on it. So I don't think we can just do it if we want. We need to get a permit first.
We do have a permit for rice blast, so just do that too, do that experiment.
Sorry.
Okay, any other questions, any of the students? You don't have to ask a QE type question. Just say quick.
Are there any rice lines with XA3 and XA21?
Yes, I think because during the during the cross we have the lines have both XC21 and XC3. Yeah.
Is that, are sort of a general question, are you pursuing that as like a breeding strategy for resistance to stack those together?
I think actually this was already been done in the EU, like International Rice Institute. They already like stacking like XC3, XC21, XC1, something. They stacking like 5 or 6 XC genes in rice and already used in the field.
And XA21 has a broad spectrum, so it overlaps already XA3. So as far as we know, adding XA3 doesn't confer broader specificity based on the limited knowledge.
Right. Well, with the African strains are still virulent against
Yeah.
XA21
But
strains.
it's
With,
also a virulent on XC3.
they are, okay.
Yeah.
Thank you.
Any other questions? Okay, well, thank you.
that mat. You gotta move that mat outside the door.
I saw you almost
It's like everyone coming in was like tripping.